Escaped Sapiens.
Despite our resource wealth and billions spent each year on pharmaceuticals, rates of depression and anxiety are on the rise in the West. It's natural to wonder what impact our modern, sedentary, and two-order lifestyles might be having on brain development and neural function. In this episode of the Escape Sapiens podcast, I speak with Kelly Lambert, who is a behavioral neuroscientist working at the University of Richmond in Virginia. Kelly uses animal models to investigate the impact that our experiences and environmental context might be having on our own cognitive function. Her findings appear to have some remarkable implications concerning the impact that behavior might be able to have in sculpting improved mental resilience, reduced stress, and improved cognitive function. I hope you enjoy hearing what she has to say. There are some aspects of your research that appear really quite fun, and I want to get into talking about how you train mice to drive cars and chase after Fruit Loops and all that. But on the other side, there's an undercurrent to your research, which is really quite serious and real, that has to do with mental health and depression and that side of things. And so I was hoping, if possible, to start off by asking you a few questions in that direction before jumping into some of the more fun stuff. So the first thing I wanted to ask you about is it really the case that in the West today we're seeing an increase in the number of in cases of mental health problems and depression and this sort of thing? And if so, why is that? Is it because we're outsourcing manual labor overseas, or is it because we're spending more time in front of the computer screen? Do you have some sort of idea of why that is?
Lots of thoughts about it. And yes, you're correct that it's for at least some psychiatric illnesses, or most psychiatric illnesses, such as depression, the rates are increasing. It could be that maybe more people are aware and reporting this. But when we look at other medical conditions, such as cancer, HIV, cardiovascular disease, the rates of death due to those diseases, at least in the United States, and Tom Insel, the past former director of the National Institutes of Health here in the U.S., had reported this. Those deaths are going down, suggesting that we're getting better at treating this. So all the research, our knowledge, our biomedical knowledge base is increasing our conditions with that, the outcomes. Psychiatric illness is just in another category. It's not decreasing, and in many cases increasing. And I know that we're all dealing with this huge stressor right now with COVID. And our Center for Disease Control here, their recent surveys of looking at not only depression, but the number of young adults who have reported having ideation of suicide during this pandemic is alarmingly and disturbingly, any rate of it is disturbing, but it's very, very high. So yes, we do seem to be in a situation where other medical illnesses are getting better due to our research and information, but psychiatric illness, depression, schizophrenia, obsessive compulsive disorder, anxiety disorders, not so much. So your question is, why? We have all these wonderful, smart people in psychiatry and neuroscience and biomedical research. That's a good question. One question, I know that you're going to ask me about rats, and I'm perfectly happy to be critical of my own field and our own work, and I think we need to look at what we do with a critical eye, because at one level we want, we're doing this for models to try to improve conditions for humans. We want basic information about life in general as well. But when we're looking at translational research, so from our rodent models, and I work mostly with rats, to trying to figure out causes of mental illness that fits in the psychiatric illness category, like depression, where are we going wrong? So we're doing a lot of research with these rodent models, but I think that we're limited by keeping we need control in the laboratory. So we keep it with proper care of animals, but they're in a very kind of predictable, standard, safe environment. Animals didn't evolve to be in that predictable, safe, never-ending resources always dropping from the sky kind of environment. So if we're looking at a brain, and a brain, we're not born with the brain, if we're looking at humans, with the brain that we have now. We have the brain that has been carved and sculpted and engineered by our experiences. So if you limit our experiences, then you've limited the brain. Same thing for these animals. If you limit, surprise predator exposure, or this new thing, or a thunderstorm, all the things that come with the wild. If you limit all that, then I think we're not only looking at a simpler model than a human, but not even an authentic rodent model. And that's something for me to say. I've been in this business more than 3 decades, and I've learned a lot from rodent research. I wrote a whole book, The Lab Rat Chronicles, about their wisdom, and we need that control for some of our techniques, but I think that has limited us. And what we see in the lab in this very limited situation, standard control, is not translating to humans. And humans, we have a very impressive brain, and we can talk about differences in brains between humans and rodents and other animals. So that aspect of the research, we're trying to uncover information from our wonderful laboratory, our controlled, systematic laboratory research, in some ways maybe limiting the outcomes because of the behavior part. So I think we really need to respect the environment. I work at an undergraduate institution, so I work with undergraduates a good bit. I'm always in class talking about context matters. So it's not just having a brain, but it's that environment that we put our brains in that is also incredibly important. So I think the research on one end has been an issue. I think that we've been limited and maybe a little just limited in our toolbox as well. We get real excited about drugs, a pharmacological way to treat mental illness, psychiatric illness, and for good reason. Going back just the middle of the last century, we were still, putting people in horrible institutions, and people were getting frontal lobotomies, and the treatments were horrible. So the idea that you can change our neurochemistry by taking a pill, it was just wonderful. We wanted to be able to do that for our loved ones. And for some conditions that seems to be working okay, but we've got a multi-billion-dollar pharmaceutical, industry for mental health. And I just said that our numbers for depression are not getting better. We've got antidepressants everywhere. So I think that we've gotten very excited, and we've just stayed on that track. And I've, just to kind of make this more noticeable, I've made up this word behaviorseuticals instead of pharmaceuticals. And that's the idea, that we can change our chemistry, neurochemistry, dopamine and serotonin and adrenaline and such, noradrenaline, through behavior and through emotional, situations, in ways just as we could, that may be more, I think, more authentic and maybe more relevant than changing our neurochemistry with a pill. Because we really haven't shown, like with depression, there's most of the literature is about serotonin. We know that more neurochemicals are involved. But drugs are your standard off-the-shelf kind of antidepressant is going to increase the activity of the serotonin, this neurochemical, in some way. So we thought, or others thought, that, we must have, people who are depressed must have a deficit of serotonin. So let's do something to increase serotonin pharmacologically, and we'll solve that problem. But we never have really shown in a very definitive way that people with depression have low serotonin levels. They're not going in and getting some, and we don't even have the technology to be able to do this in a way with the public and living brains. They're going many times into a general practitioner saying, I saw the commercial about the symptoms, I have this, and they're in pain, and then they write a prescription. If someone has diabetes, the doctor just doesn't write a prescription for insulin. They're going to do blood tests and look at biomarkers. And so we don't have easy access to biomarkers. So we've assumed, and this is it's faulty reasoning to think that a correlation is causation, is what we teach our students in the 7th grade, right? So, or this indirect way to, that indirectly serotonin is being increased, but it doesn't mean that that increasing serotonin is the cause. I may have a cold and eat my grandmother's chicken noodle soup and I feel better, but that doesn't mean I had a chicken noodle soup deficit. There's something about that is doing something that may be making me feel better. So it means that we still need to be more, better brain detectives about, okay, why are these antidepressants working for some and not others? And why are we not seeing overall better outcomes when we're relying on the drugs? So everything's complicated. We need to take that knowledge that we know about the pharmaceuticals and still respect more behavioral cognitive therapies, which have very high efficacy rates. It's not as easy as taking a pill. We'd love to have a pill to lose weight. Nobody wants to have to exercise and eat the right things and diet. Our brain's kind of the same way. We probably need the hard work with that to change the neurochemistry in healthy ways. And so maybe we could add behaviorseuticals to the pharmaceuticals and maybe start with behaviorseuticals when we can. So a limited toolbox, limited research opportunities with these models, I think is contributing to it in a different way than we're seeing with other medical conditions.
This is one thing I wanted to ask you about. So when we're trying to address depression with pharmaceuticals, is the model then that there's only one type of depression? Or do we know there are multiple different types of depression that correspond to different chemistries in the brain?
Well, there's certainly bipolar emotional disorders, bipolar depression, which seems to be very different than what we call this unipolar or major depression. And then there are different degrees, Shane, of milder depression leading to depression that may be associated with the suicidal ideation or death by suicide. So there's certainly those degrees. But there's not a sensitivity of, even seeing that someone has a deficit of serotonin, and so we certainly don't have, well, you have mild depression, then let's give you, there are different drugs that are involved, but we haven't really been able to be that sensitive about, okay, this particular cocktail or drug is going to be best. A lot of times, because the drugs are different in some ways, and I should state that I am not a clinician, so I need to be very careful. I'm talking to you about my research being a behavioral neuroscientist working with animal models. But a drug would be tried at a certain dose, and then when that doesn't seem to be working, then another dose is tried, or another drug, or another combination. It seems to be more hit and miss than we should be.
Which seems quite dangerous when you're dealing with suicide as well. And
yeah, I think it is. I try to, I'm not saying at all that people who are taking any type of medication should stop. I'm not saying that, because if you've been taking medication, your brain has adapted to that. That's part of the wonder of neuroplasticity. And so the last thing you want to do is stop. So if you've been taking a drug, your brain has adapted to having more of that, so it may have cut back and compensated for something. So if you just automatically stop taking it, you're going to be disrupted. But it is important to try to understand what's been done, to ask questions to your doctors, and there could be some real danger to being too dependent on disrupting natural neurochemistry with pharmaceutical agents for long periods of time. When does our brain stop knowing when to produce certain neurochemicals and to change? And are we just kind of in this I don't want to be too dramatic, but a chemical cage of being dependent on taking these drugs to feel normal or average? And we're not talking about addictive drugs, for other emotional outcomes, recreational drugs and such that can be very addictive. But some of the medicinal drugs can be too, because your brain adapts. Well, not that it's medicinal with alcoholism, but people who consume alcohol, that's a suppressant. GABA kind of suppresses activity. So your brain compensates because it doesn't want to be suppressed all the time. So it's going to compensate by gearing up some of the more excitatory neurotransmitters. So if that person stops drinking alcohol and the brain is addicted and used to the alcohol, that person's going to feel fidgety and nervous.
And it can actually be quite dangerous as well, right? Coming off alcohol.
Yes. And there could be drugs that are excitatory, the compensatory is the other way. And so overdoses or people who take drugs outside of the context that they're used to, their body may not compensate in certain ways. And so a dose that was perfectly fine in this context could be an overdose and lethal in another context or combined. So you're right. Pharmaceutical agents of any kind should be taken very seriously. And I think that maybe we've become too dependent in the psychiatric realm on that, even though our neurochemistry is very important to our functioning. So I'm not trying to downplay the role of the neurochemistry, but we can certainly change our neurochemistry through behavior, through emotions, through cognitive processes as well. So that should still, I think, be on the table and be an option.
No, so yeah, that was one of the things that excited me about your research. So you're looking at alternative ways to address some of these problems that we have. Can I ask, you're dealing with animal models in particular. You're working with rats and looking at how behavior will affect their stress levels and anxiety and this sort of thing. How good are rats as a model for humans? You mentioned that they have very similar brains. They're smaller. But for example, are the neurons the same? Is the chemistry the same?
Yeah, great question. And my thoughts are evolving a bit over the last couple of years. So for the longest, I've said, and I still believe that a rat is a very good model for a rat brain for the beginning stages of research. So what you're asking, neurons or the cells in the brain look exactly the same. If I've down the hall in my lab, if someone puts up puts a slide with a human neuron under the microscope or a rat, if I don't know the content or I can't see the size of the slide, I don't know the difference. So the structure of the neurons and the glial cells, the supportive cells are the same. Generally, we have all the same brain areas: a cortex, a hippocampus, a cerebellum, an amygdala. Neurochemicals are the same. We share a lot of neurochemistry with plants even, so there's been a lot of conservation with chemicals throughout evolution. Rats, I'll be the first to say, they're very complex in their behavior. They teach me things all the time. I'm learning from them. So just like other biomedical researchers may start by looking at cells in a petri dish to understand how cancer cells may react in a human body. This is certainly a great model. We have an intact brain with all the neurochemicals and behavior. That's a great place to start. But the human brain is very different quantitatively and maybe qualitatively. We're still on a continuum with evolution, but it's a numbers game. So one of my colleagues, Susanna Herculana Huzel, who is at Vanderbilt, but most of her career was in Brazil, where she's from. So she came up with this wonderful technique, innovative technique to count neurons. So these are how many cells do we have in our brain. So let's look at the human brain now. So if you went to movies and such, there's 100 billion neurons are in the human brain, and you kind of, okay, I'm not going to count 100 billion. How do we even kind of know 100 billion? And it's a sampling, they would take tissue and count. And so Susanna is wonderful. She was working actually for a science museum in Brazil, and she wanted to have some of these fact sheets, and being a good scientist, she wanted to go back and check the literature. Okay, how do we know that humans have 100 billion? Because we know that we can't really count all that. And it was kind of like, street lore or a folk wisdom or something. It was informed, but no one had really, there wasn't a definitive original reference. It's kind of even in textbooks. Well, this textbook said it, and this textbook said it, and I write textbooks myself, so I know it's hard to track down all this. But it was when I was writing a textbook that I needed to find out. And so she came up with this way of homogenizing. So the challenge with counting cells is there are different density of neurons in different areas of the brain. So you just can't go, oh, the brain weight of this, let's extrapolate. You need to look at the density in different brain areas. So she pretty much had a blender of sorts, and she talks about making brain soup. And so you like take the outside area, the cortex, or that back area hanging off the cerebellum, and you mush it up, you make a soup, and it has the cell bodies that are still intact. And so you add volume, you have the weight of your brain, and then you add volume, and then you take a small sample, and because they have dispensed, in equal way, you can start to extrapolate in those different brain areas. And she could do that with a small brain and really test, validate this. So when she used that, and that's called isotropic fractionation, when she used that technique of homogenizing and counting the cells in different brain areas and counting it up, so her estimate is 86 billion for a human brain. And you may say, well,
that's not so far off.
Yeah, that's what you say, but 14 billion is an entire baboon brain. So, I don't know what an error of,
So we just became dumber using a blender.
Well, yeah, if you define intelligence by number of neurons, we became more, maybe we became dumber, but became more accurate. And then, but using this technique to compare to other brains, I think we still are impressive. So that's the point I'm trying to make. So using this technique, she's looked at a lot of different species. And even though we don't have the largest brain, elephants, dolphins, and such have larger brains, we have more neurons in that cortex, the outer area. We don't have more neurons than an elephant, but we have more neurons in that outer cortex. And primates in general, when she works with the numbers, they scale at a different level than rats do. So if rats, going back to, is rat a good model? If as rat or rodent brains get bigger, their neurons get bigger. And so they don't densely pack, they don't change the density as fast as primates. As the brains get bigger, the cells stay the same. So they're really increasing the density of neurons. So you'd have to, there's a big rodent called a capybara, and even that, it's just more sparsely counted neurons. So, so there are a lot of ways to compare brains, but I think a rat has about 200 million neurons generally compared to our 86 billion. And then when you look at a human, even more so than the other apes, orangutans and gorillas and chimps, we've got this really impressive number of cortex neurons. And I know if you're kind of thinking about with Hamilton, they talk about the room where it happens, that play here in the US. The cortex is kind of the brain room where it happens. There's so much specialization, integration, fast projecting cells, regulating our emotions. So that's a difference between the human and the rat brain. And because we have this processing potential, then you get into questions about, memories and anxieties, the way that we have them. And, we don't have to be, one of my favorite neuroscientists is Robert Sapolsky, and he has this book, Why Zebras Don't Get Ulcers, was one of his early, really popular books. And
do they?
Well, he says they don't really. The point is that zebras, they get really stressed out when that lion's chasing them. and for good reason. And stress is a wonderful response in the short term. They're able to, outrun, hopefully, the lion and such. And then the stress is over. But something that we've inherited with this fancy schmancy cortex and all the neurons is we don't have to be encountering stress to generate it. So we're perfectly happy to go, well, sometimes even when things are going well, I'm thinking, oh, what am I missing? I'm not feeling nervous. Certainly there should be something I'm worrying about. So we generate this anxiety. We have ideation. We think about the future in good and bad ways that we probably are not replicating in a rat. And psychiatric illness in many ways is about, delusions and anxieties and dread. And the psychiatric illness may very much be, somewhat specific to the human brain. Stress is not. That's been conserved. We can study stress and learning abilities. And I think that what we're doing with those models is still very informative. We can do things to disentangle information and variables in ways we can't with the humans. So we've got a huge difference in numbers. So a lot has been conserved: brain cells, brain areas, neurochemicals, basic design, universal design, and that's something that we're trying to look for. What are some of the universal designs of kind of behavioral neuroscience, the brain, and behavioral outcomes? But there are the numbers difference. There's another cell that we're looking at in the brain, a shape of it's a type of cell that just in the last couple of years has been on my radar, and that's called the von Economo neuron. And so this is a neuron that's it's a spindle, it's very skinny, and it's in certain areas of the human brain, one involved in emotions called the anterior cingulate cortex, and another incredibly interesting area called the insula that is processing information about both our internal and external environment. So it's so important. And they're there. And at first we thought, oh, they're only in apes, and that's kind of what makes us special. But then we found them in pigs and horses and cows. And my new species that I'm just wild about and I think we can learn so much about is the raccoon. I don't know if you're...
I used to live in Canada. They used to be in my roof, so I know what a raccoon is.
And for all the reasons people don't like them, I love them. They're problem solvers, they're persistent, they seem to have kind of this curiosity. So we've been looking at raccoon brains that we're inheriting from USDA or wherever when they have
And they also have this complexity.
And they have, they have the von Economo neurons, at least in the insula. Rats don't have them anywhere. I've looked and looked and no one has found them. And the raccoons, and this is some work done with Susanna Herculana Huzel as well, their cortex profiles like the primate cortex. So there's the existence of von Economo neurons. So there are some subtle, and also the cortex, if you look at our cortex, it has all these bulges and wrinkles, and because we've got to put this flat sheet of tissue into this small head so that the females can give birth to this, can be this flat. So we've got all these wrinkles called gyri and sulci, and the rat is just smooth as a baby's bottom. There's no, we call it gyrification, but the raccoon has this. So again, I think we can learn a lot from the rats, but we need to consider looking at some of their wild behavior, natural behaviors, and we need to look at different animal models. There's not going to be one animal model that's going to be the best, perfect, that we should hang our hats on. Today in biomedical research, the rat is gone, is the mouse, and the mouse even has a simpler brain and behavior. And no offense to the mice out there, I've done some wonderful parenting work with the California deer mouse, and they're interesting and very, mice are very interesting and survivors, but the rat has a little bit more complex behavior, I would say, from my experience with them. So there are limitations to putting all of our eggs in the rodent basket for biomedical models. They're very valuable, but we need to realize the limitations of these and try to branch out to see where we can make a difference. So much suffering going on in the world.
So despite the limitations with working with rats, I still wanted to ask you about some of your research in that direction. So you have some research where you're looking at the environments that rats are living in, where you have to correct me if I'm wrong here, but you have sort of country mice and city mice and these comparisons. Can you sort of explain, how do those experiments work and, what are you looking for?
Right. So in my lab, I kind of describe what we're doing as looking at experience-based neuroplasticity. So neuroplasticity is this wonderful aspect of brains that allow them to change, from the womb to the tomb. So if our environment changes, our brains can change with them. So some simpler organisms, it's more reflexive. If the environment changes, they're toast. They can't adapt to this. So back in the 1960s, here at Berkeley, University of California at Berkeley, there was a team of researchers who did some fascinating research on what they called enriched environments. So they were able to look out of the box, or the lab box, where animals were kept in these streamlined, very clean, predictable environments, and put them in a messier environment. They called it kind of a Disneyland, where there were new what we call stimuli or objects put in there every day. It was a larger enclosure, so they had more physical movement. They had more animals in there, so like 6 to 10 with a larger space, whereas a lot of research may have an animal just housed individually, housed. So after only about a month, they noticed when they looked at the brains that there's more neuronal complexity, such as for the existing neurons, there are more branches, and neurons are wonderful in that way that they communicate with so many synapses.
The actual morphology changes, not just the chemistry.
Right. And so this is part of, neuroplasticity could be the production of new neurons. That takes a lot of energy, and that can happen. Or the neurons you have can make new connections. And so that's very much a part of what our neuroplasticity as adults probably is more about, making new connections and more, faster connections and circuits that are building. So they started to see some positive brain changes, and that was considered an enriched environment. So in my lab, working with undergraduates and being interested in neuroplasticity and environments, we started to look again at that. And we were asking, when we were looking at the protocols in the scientific literature, a lot of the stimuli, the enriched stimuli, were kind of artificial mirrors and metal things and things from little Happy Meals from kids. And they weren't very natural things. And so I thought, well, I don't know, does that make a difference? Is it just novelty, or is it something about where, this object came from? So we started to build what you're referring to, these what we call natural enriched environments, which could be kind of like a country environment. So we would have sterilized dirt that they could actually dig in, as opposed to the more manufactured bedding. Instead of a plastic ladder, we'd have a stick. Instead of a little plastic cat ball, we would have a rock. So we would control for the function associated, potential function, and the number, but we would have kind of an artificial and a more natural environment. And the person, Marian Diamond, who's one of my heroes, who was in that original 4, one of the first, unfortunately, there aren't a lot of women in neuroscience that you're reading about in the 1960s. But she was still alive when I was going through this. She passed away a couple of years ago. And so I was able to interview her, because I love interviewing people to get that story, because I was working on a book, a textbook or something. And I said, why all the artificial? Did you ever look at natural? She said, yeah, we tried that. They had like a sandbox or something at Berkeley outside. And again, it's hard to control for things. And so they talked about it in a Scientific American article, I think in 1972 or something. They had the cover of Scientific American. And she mentioned that the animals that were outside in this more natural enriched environment had heavier brains or something like that, but it wasn't systematically published. She did mention
they didn't blend the brains though, or use any of those more modern techniques.
Or they didn't have a lot of animals in that. but she was encouraging to think about this. So we tried to bring nature to the lab instead of taking the animals outside. And so overall, and we're still, every study, we add to this, but overall, we think that regardless if it's the artificial city or the natural country, adding new things to the environment and having the ability to move and social interaction is so rich. You never know what this other animal is going to do. That seems to be good for their cognitive. Well, the animals had similar cognitive abilities, learning abilities. But when we looked at their emotional responsiveness, like giving them a situation, they don't love to swim, but they're great swimmers and they will swim in nature. But, look at their stress hormones when we expose them to a challenge task or something. The natural animals seem to be more what we call emotionally resilient. So their stress hormone.
How do you measure that?
Yeah. So one way is to look at stress hormones. So we have the stress hormone called cortisol. You may have heard about it. It's not great for it to be high for long periods of time. We look at the rat version of that, a corticosterone. And then we've also been interested in this other, that's released by our adrenal gland. I'm pointing to my kidney here. It sits on top of the kidney. Another adrenal steroid, DHEA, it has a long name. It's also, that's considered to be a buffer to cortisol or corticosterone. Even in humans, some of our special operations studies that have looked at highly trained soldiers, special operations and such, they have a higher level of DHEA, the more successful and trained ones. So there's a good bit of research. So we like to look at this ratio because I think ratios are more informative than
so if your DHEA is higher, that means you can deal with stress better?
Perhaps we think that it's less toxic. And so we're able to look at that. You could look at it in the blood, but it's we don't like to, it's hard to get blood from a rat, and it's a stressor in itself. So we have a technique where we get it in their poop, and they're perfectly happy to give us poop. So we can assay out these hormones from fecal matter at any time. So you can look at it that way. You could look at certain receptors for stress hormones and such in the brain, but we've mostly been looking at those fecal-related hormones and looking at behavior itself. So we're still looking at more sophisticated brain ratios or receptors and such. But we think that something about that natural environment may be differentially influencing the stress response. And that's not a big surprise because even there's research with humans that a walk in nature can reduce stress hormones. And there's this wonderful correlational study in Denmark a few years ago. People who grew up with green spaces outside or close to a park, or I forget how they measured it, shrubs in their front yard or close to a park or something, were like 50% less likely to be diagnosed with psychiatric illness as an adult. That's huge. If we had any drug that could reduce that way, it would be so great. So we know there's something about nature. I mean, our brains evolved in nature. So it's just interesting to be able to pick this apart a little bit in the lab. So it's enriched can take on a lot of different meanings. We're kind of leaning more toward engaged. And if we're talking about lifestyles that are healthy, engaged, a wild rat that is in nature, and we're starting to look at wild rats, it's not Disneyland. It's very stressful, maybe like our lives. So we need to be careful when we say enriched environments are just like the wild. No, it's enriched out there. There's a lot going on. They have to be on their little tippy paw toes or whatever. But there's predators and poisons and all types of things. A rat in the wild probably is rare for it to live past a year, maybe not past 6 months.
How long do they live in
the lab?
Good conditions? Yeah.
2 years, maybe 3 tops.
That's pretty impressive.
Yeah.
The difference.
Yeah. So if we're looking at really different animals, a wild natural animal and a lab animal, that could contribute to our findings. So it's just something I think we need to be aware of.
So it makes me wonder, drawing lessons for ourselves. Some days I'm sitting in front of the computer 2, 3, 4, many hours, right? And what am I doing to my brain?
That's a good question. We're all in this experiment that I'm not sure we signed up, we gave consent for, but we're doing it. I'm in that situation as well. One of my areas of research that's kind of related to this, so I'll bring it up because I think it's related to what screens may be doing. And trying to look at therapies that may not be pharmaceutical related and more behavior-ceutical, we have some research that we call effort-based reward training. So we're looking at animals that get their rewards, which is in my lab, a Fruit Loop. It's not very natural, is it? But they like Fruit Loop, this little sweet cereal. Do what we can. But we don't just give it to them. They're put in a little arena that they're familiar with, and we teach them that if there's a mound of bedding, that if they dig down with their little paws, they can get this Fruit Loop. So we're not talking about heavy duty work. It's 5 minutes. There are piles that move every day, and they go and they dig up this Fruit Loop. So I'm trying to build connections between effort and reward. And when you have connections between effort and reward, maybe that increases a sense of control. I know how to get the Fruit Loop. It's not just random. Or this maybe more human word, self-efficacy. I've got this. Have some control. So we compare several measures of these effort-based reward trained, and it's contingency training. The reward is contingent on their effort, to animals that we just give the reward to. So the contingent trained animal dug up 3 Fruit Loops, we would just give this control group 3 Fruit Loops. We put them in the arena, but there's no contingencies there. They just get it. So we call it the trust fund rats is what we call that. So they get the same number of rewards. They kind of have a similar experience, but we're not building these circuits. And so we also see emotional resilience in those effort-based reward animals. So having a sense of control and our brains, most of our brains are built to move us around. I mean, we get all excited about this decision making and intelligence and such, but that big part in the back of our brain called the cerebellum, in humans, about 78% of that 86 billion neurons, they're in the cerebellum, and that is involved in motor coordination and movement. So healthy brains need to be moving, and they need to have some sense of control, self-efficacy. When we're sitting in front of a screen, I'm moving my hands, but I'm not traveling in any way, so my movement is limited. It's just less engaging for the brain. If I was having a conversation with you, it would be more, in real person, it would be more engaging.
Of course.
So, and also as our world, just over the last 100 years has become more automated and westernized, technologically advanced, we've lost control of being able to change things. When I was stories of like the first cake mixes didn't require you to do anything but add water. But I think there's some controversy. There's not scientific literature on this, but there was some writings that they figured out that if the bakers, mostly women in those days, if they had to add an egg, if they had a little skin in the game, they were more proud of it. They had more of a sense of accomplishment. And so when everything's automated and we're not moving around a lot and our senses are limited because it's just focused here, I don't think that's great for the brain. I think we need to be mindful and at least have add in nature or hobbies or things like that that can remind us, the way our brains and bodies evolved.
So a quick question with regards to this is, in America, I'm not American, but in America, there's a huge prison population, right? So what's that doing to those people? When you're locked away, you have zero control over what's going on. You get your meal every day. You have very limited input. Are we creating a situation where we're going to have huge amounts of depression and mental? Are we generating mental problems in these populations?
It's such a good question, and I've just reached out of my kind of comfort zone with animal models to write a collaborative grant proposal to look just at that, prison populations, because based on everything I know about the brain and behavior and nervous systems, I think you've taken individuals who are vulnerable, whose behavioral output and decision making has not been great, and you've put them in the worst situation possible for that brain to improve. It's like putting someone with lung cancer into a highly polluted environment. There's value judgments about what people deserve. I don't want to get into that, but if we want to rehabilitate, if we want to be able to send someone back out into society to be a productive member and to be safe and not get into trouble again, the way that we see prison today, in my mind, is like those little stripped down, impoverished environments of the rats in the lab. And if we're going to be concerned about that, we certainly need to be concerned about humans. If you talk about or think about solitary confinement, that has to be just torture for a brain. That I don't even know if a brain can recover completely, depending on how tense and chronic that is. So you're right. I've been talking about neuroplasticity in more positive ways, but our brain is changing when it's impoverished, and we can talk about poverty and low socioeconomic status and such. But I don't think that's the smartest way to try to rehabilitate a brain. I'm certain about that, and we need to look at that in more responsible ways. And you're right, the U.S. we're being the most irresponsible, I think, in many ways about that. We could do better.
Mm. So in the experiments that you're actually doing, when you have an enriched natural environment, or when you have rats that are working for their Fruit Loops, so you're saying they tend to have greater cognitive abilities and greater, not in terms of the environments, but for the working rats, they have better cognitive abilities. Is that the case?
Yes. So for the working rats, so several studies, what we did with those is after we train them, and it takes about 5 to 6 weeks to train them. And a lot of times with rodent research, we don't love long training. We like to do something fast and then see what the outcome is. So after we've trained them with that task, if we give them a new task, like one that I've written about and published, is this little cat ball where the Fruit Loop is in this little ventilated plastic ball, and they try to get it, but they can't reach through. Their little paws can't get in. And I love to see an animal, and even a student, when they're trying to figure something out, I love to see it. So they'll kind of put it in their mouth and sling it against the wall of the cage to break it out, but nothing works. So occasionally they will figure it out, but we've tried to rat-proof it where it can't be opened. And so what we found is that effort-based reward animals would persist longer before they gave up. And there's a literature in psychology, which my training is in, called learned helplessness, where it was originally done with Seligman and with dogs and rats, where there was something they couldn't escape from, like a mild
This
shock.
is the electroshock.
Yeah. They would just give up. It was like, I can't, nothing. And that seems to be a perfect model for depression, where you just give up. No matter what I do is going to help.
Could you maybe explain those experiments a little bit? Because I'm not really familiar with them. How does that tie into what? Sorry, I'll let you go.
Yeah, well, it does tie in. So what they saw, they put the original research, 60s or 70s, dogs were in a 2-compartment apparatus, and there was a barrier, and so they were in this one compartment with a barrier, and there was a very light, I think, shock on the grid of the floor. So it's uncomfortable for the dogs, and usually a dog would try to get the heck out of there, but they had the barriers high so that they couldn't jump out. And so they did that a time or two, and then they dropped the barrier down, and they did it again. And this time the dog could jump out, but they saw that in about I think 70% of them, they just kind of sat there and took it as if they had learned in the previous trials that no matter what they did, it wasn't going to change the outcome of the situation. And so they called that learned helplessness. And it seemed to be a very good model or way to think about depression. You don't get out of bed because no one's going to hire you. No one wants to be in a relationship with you. That has an interesting outcome with Martin Seligman. And later on, he said he was giving a talk to psychiatrists, and he said, 70% of the animals are engaged or showing or exhibiting this learned helplessness. And someone asked the question, what about the other 30%? Yeah, that's fascinating that even after being taught that no matter what you do, that you still go for that. And so he kind of shifted and wrote books about learned optimism. And that's very interesting in itself. But I think what we're looking at is maybe a more learned persistence, as if in the training they learned that their efforts could result in reward, even though they had sometimes they messed up or they had to keep at it. That maybe that generalized, so that they with given a new task, and that's what the true test is, that they can persist longer. More recently, we've been looking at a different type of cognitive ability called pattern discrimination. And to be informed, you need to be vigilant about your environment and notice, oh, this job's not working out very well, or this stock's not doing well, or my kids are acting out, noticing when patterns are changing. And that has been used as kind of a model for not being attentive to the surrounding environment could be important for something like depression. So we're still in the midst of analyzing this, but we noticed that our trained animals, when we gave them a little like Lego designs that were different, a certain percent of the blocks had been changed, that they could notice the more subtle differences, and that we study by how long they're kind of gazing at it. If it's new, they'll gaze longer. So being more attentive in the environment, because you need to look at where the Fruit Loop mounds are, could be important as well. So something like depression is so complex, but having an engaged brain that is still changing in response to the environment, not giving up, not resting, in maladaptive ways, being attentive to things that are happening in the environment, having a sense of confidence that you have some control over the environment to gain resources, is incredibly, I think those are important. And all of that can depress chronically high stress hormone levels. If I want to find a bad guy, a common denominator for so many psychiatric illnesses, chronically high stress hormones would probably be way up there. We do know that we don't know that serotonin is a little higher or lower, so to speak, with depression. But in about half of individuals diagnosed with depression, they have high levels of stress hormones. And that is something we can measure, in blood and saliva in humans quite easily. It may be a biomarker that we could start to look at. And another reason, going back, I know I'm going all over the place, Shane, but going back to your question about depression, why is depression getting higher, and the other medical conditions not, we have some good biomarkers of cardiovascular disease and cancer and screenings and such, so that we can attack it before it gets bad. We really don't have those biomarkers for depression or schizophrenia. So it's not until someone has a mental break, a depression episode that's very dangerous, maybe even a suicidal attempt, or a schizophrenic break, attack or episode, that we start to treat it. And if we had known years before, then maybe we could have kept that from happening or from being so severe. Same with neurological diseases, Parkinson's disease and Alzheimer's disease. By the time someone's diagnosed with Parkinson's, so much of the brain structure called the substantia nigra has already degenerated. So that started happening years before. So if we knew it then maybe something could be done to buffer that. So we need better focus on, or just better information about biomarkers so that we can start to address this before, if we could only treat people who had cardiovascular disease after a heart attack, we would not be as successful in diminishing the death rates due to cardiovascular disease.
In terms of Parkinson's disease, how do we know? So if we know that that it had started many years before, what are the biomarkers that tell us that? As in, do we know what the biomarkers are? Are there tests that we can do?
Well, I think again, I'm not a neurologist, but I think that most of that has been done with brain imaging, so that you start to see the structure diminishing itself.
I see. It's actual morphology.
Yeah, and then you can look even at autopsy of people who may have died from different reasons, but at different stages. Mm-hmm. But it's a longer process, and Alzheimer's too. To the
so that might happen for years before you.
So you see a symptom that is diagnosable. Mm-hmm. That's a lot of room and time that maybe now I say that we don't have that intervention necessarily now, but unless somebody was going to focus more on behavioral training, there's a lot of brain training that may be able, or behavioral cognitive therapy, or different types of training can also build buffers. We're still trying to figure out the efficacy of all that.
Going back to the learned helplessness that you spoke about, I'm curious about, what might be the evolutionary benefit to that? Could it be the case that, say I have an injury that will never heal? Is it a good thing that I just sort of come to accept that? And where does learned helplessness actually come from? Is it some function that's broken, or is it, how should I understand where it's coming from?
I think it's probably a very adaptive function, and it may be related. If you look at sickness, we've been looking at the immune system with the COVID crisis. When we get sick, kind of regardless of what you have, when there's an immunological threat, our symptoms are very similar. We're kind of achy, we don't want to get out of the bed, our appetite may be influenced, got kind of these fevers or whatever. So we're not, we're kind of limited in what we're doing in order for our body to heal, for our immune system to attack whatever is the threat on board. I see the stress response and anxiety as a psychological extension of the immune system. So if something's not right with a family member or a loved one, a work situation or something, maybe you need time to regroup, to consider that, to think about it, to be sad about it, to process it, so that you can figure out what is not working, and then figure that out, and then maybe life is better. But just like if we were sick and our immune system's trying its best to work overtime using energy to address an immunological threat, it's probably not good for us to be out, our ancestors traipsing around, and expending energy, making ourselves more vulnerable. We need to focus that energy on healing. And so I think it could be very adaptive to have times where you're regrouping, where you're questioning how things are going. And sadness, we've kind of medicalized sadness, but I think emotions, the whole, I like to think of healthy emotions kind of like this quilt. You have all these different patches of different things that make it richer. So sadness can certainly turn into something more pathological, but the learned helplessness, where you're not doing the same thing, in the sense that it may be kind of like a sadness, if it's transient and temporary, where you're not going to keep trying the same thing that didn't work, that could be beneficial. Now,
saving resources.
Yeah, so it could be related to that. Or it could just be this learning, it's a limited learning where you've learned that the dogs learn no matter what I do, it doesn't work. So it's kind of a limited learning, because there's obviously something else when the situation has changed that the animal could do. But if stress is limiting our scope, our pattern, that's a pattern that the dog didn't see. The barrier's lower now. I can probably do this. Let me recalculate. I think the brain, my recent book talked about contingency calculations. It's one of the big purpose of the brain, to keep calculating contingencies as the environment is changing. So a stressed brain is going to be less likely to be able to do that. But as it's related to kind of sadness and kind of staying within to figure things out, that may be adaptive. So I think there's an adaptive and a maladaptive aspect to it. And if it's chronic and it goes on for too long, it's all going to be maladaptive, because you're not going to be able to get the resources that you need, and stress is just going to continue to expand, and that's not good.
So on the side of neuroplasticity, this is where your research with rats driving cars comes in, right? So you've trained rats to drive little miniature cars around in search of Fruit Loops, and well, maybe you should. So you're trying to see whether rats that have the worker rats as opposed to the trust fund rats, which ones do better in their driving tasks? Which ones get their licenses first?
I just lost a lot of credibility when you said little rat cars, and so a little bit. No,
that wasn't what I was trying to do.
It's kind of fun. Again, I'm with undergraduate students, and just the backstory to that is, I had a colleague in cognitive science. I came to the University of Richmond about 3 years ago, so I had new colleagues, and she was in cognitive science, and she had seen some video of a museum that had an aquarium with goldfish in it, and where the goldfish would swim or school, it would move the aquarium. Not that they were consciously aware of that, but it was intriguing for her. She's very bright, always thinking about innovative things. And she saw that and she said, "Kelly, do you think you could teach a rat to drive a car?" And of course I just laughed at that. Why would I want to do that? I'm this serious researcher looking at these other areas of neuroplasticity. But, I thought, well, I'm kind of new here. Let me be a team player. And before I knew it, we were having coffee and talking about these rat cars and designing, how would they activate it and steer. And it just became very fun and engaging for our brains. And it's a learning task. And I started to appreciate some of the value of having a complex task. And where they're steering, I'm boldly calling it a skill, because we could put them anywhere in the arena. So they can't just learn turn right, left. They need to kind of figure out where they are and then navigate and turn that.
And they can do this.
Yeah. I mean, it was totally, she was building the cars. I was the animal.
Were there multiple models of cars? Did you start off with some models that just didn't work? They couldn't control them. What was the
so
process
we started
like on
off,
that side?
a couple of things. Everything's a learning experience. So at first I thought that they would, if she was going to have a metal plate and they would need to touch. There's no electrical, no subjective, no feeling of this, but if they could touch a wire and complete a circuit, it would activate the car. And I thought, well, rats are very olfactory dependent, and they're always sniffing and smelling. So I think, based on my 30 years of rat expertise, they would touch the wire with their nose, and then that would close and then they would. And so that wasn't working. Like, darn it. So we started off, the cab of the car was just like a plastic cereal container, and it had a lid on it, and we started off without the lid. We wanted them just painstakingly slow shaping to, here's the Fruit Loop, and to get in on their own accord and everything. So it's not very restrictive. It's still a big plate kind of area. So one day we just put the lid on there so that it couldn't jump out to see. And I think it tried to get out, and it tried to get out by grabbing the wire and kind of reaching like that, and it drove it that way. And then I knew, okay, grasping is going to be a better way for them to activate. So she started to design the car that had this little wire that it would grasp, and it would do it this way for left, and it's got.
So no levers.
No, well,
That's too much.
Then she wanted to try a joystick kind of situation, and so we have some funny footage of they would kind of put their foot on it and just kind of pedal to the metal, and they just didn't have a lot of control. It was too wobbly. So that's was out. So we went back to the steering, because when we wanted to steer, we thought they could, turn it that way. So then we had to go to, okay, we're just going to put the wires on the right to steer right and the wires on the left. And then she went on, she's working with Apple now, because that was, they found these fun, her other work in cognitive science as well. But I've started to collaborate with a robotics computer scientist kind of person at Randolph-Macon College, and John McManus. And so he is, we have a dashboard with more of a proper little lever that we did, kind of like human factors engineering, but rat factors, to see, do they want to push in or down or out? And we're trying to look at a conceptual level. Can they learn to turn as easily by just pressing a lever on the left as turning their body to the left? Because turning your body to the left is what they would do to turn left. And we put a little mini cam inside these, so we could look at some of the micro behaviors, which I'm mostly looking at the back of their big head with this. So getting back to which animals can do it better, in this case, and what we published about this, the animals that were in the enriched environment, and it was, I think, more of a blended natural and artificial, they could learn to do this to criterion, where they drive to a Fruit Loop tree. So so they could learn to drive to criterion, but the ones in the standard environments, for the time that we gave them, couldn't learn to do it for that particular study. So the environment seemed to limit the way they would interact with new things in their environment and the way they could take advantage. There's a literature about affordances, and our experiences influence how we interact with stimuli. For example, in our culture, we know if something has a handle, I'm not going to stick my nose, I'm going to pick it up like this. So knowing that, I can take better advantage, have more affordances, options for use for that cup, and seeing new uses of stimuli and challenges in the world as well. So we're finding that the enriched animals can learn these complex tasks more efficiently and readily. And more recently, we've started, I'm concerned about research, in cages, because even though we know that these enriched, engaged cages or whatever, complex cages, are probably better for the brain, they're more expensive, they're messy. I don't see huge laboratories having these big cages with dirt in them and such. So my students and I have tried to come up with a compromise. And so we have a cage called rat lofts. And so they have a cage on top and bottom, they're standard size, and then a tube connecting them. And because when we go back and try to find out what is the variable, the most important part of an enriched environment that keeps their brains kind of more active or is healthy, travel, the ability to go from one space to another, and I mentioned that our brains are
some level of control
to move. I mean, like we were talking with the prisoners, knowing that even prisoners, unless they're in solitary confinement, they can go from one controlled environment to another. But to not be able to ever leave your environment, how does a brain that wants to process new information, how does it even exist? I don't know if mine could. So just to be able to go upstairs and downstairs, and so as we're comparing in our most recent driving study, and this isn't published and we're still working on it, the loft animals do just as well as the enriched animals. So and we're not putting new stimuli in there, it's just the ability to kind of come and go. And they do get exercise, kind of going up that little thing. It's kind of cute to see. So that may be something that's more manageable that labs can go to. It's twice the space instead of 6 times the space. But I think it's something that is becoming it's going to be harder to ignore. We need to look at the context of these environments.
This sounds like it might be something that becomes quite important for other studies, right? I mean, if you're doing any study on a rat and that rat has been completely constrained for the entirety of its life, how can you draw any conclusions?
That was my point about the limits of psychiatric research that is about the brain when we're using an animal that has a very impressive proper brain, but we're keeping it in this limited environment. I'm concerned about that.
One thing I wanted to ask about is, so you seem to use a lot of Fruit Loops. Have you thought about looking for funding from Fruit Loops? I mean, the reason why I ask is because I imagine when you were first writing the grant to build cars for rats, how do you write a grant like that? It's a very serious project, of course, but
you don't, Shane. You don't. It would never be. I mean, when I was getting into it, I knew that would never be funded. It's too outside of the box.
But yet it's gotten so much attention.
You're right. So isn't that interesting? So my funded research from National Institutes of Health with the effort-based rewards and such, maybe a few people will look at those publications. Maybe you ran across a few. But when we did this, I knew that the driving, it had this appeal. So we had the videos. It would be a wonderful platform to get students and kids, and I've heard from so many, interested in behavior and enriched environments and neuroplasticity. So our media public relations department here at the University of Richmond, they have software that keeps track of media, attention to faculty publications and things that we might write mainstream wise and such. And so it caps off at 1,500 stories. We beat that. We capped out. So it's like more than 1,500 media related stories, tidbits on these rats, which is, not as serious as all the other stuff that I've done, but it's gotten people's attention. It's intriguing. And I've tried every chance to, like I'm doing here, to say, okay, this is the science and the potential that we can learn from it. And it's very rare with rats that we do something, that the steering part that we published, that wasn't part of the enriched environments. They were in regular cages, but it took like 9 months and it was hit and miss and they were figuring out what to do. So I'm not aware of maybe any research where they've been training an animal to do something for 9 months, a rat. They only live 2 years in the lab. Our driving protocol that we're doing now takes about a month and a half. We're figuring that out better, but still, it's a more of a messier, complex cognitive design. So I understand why everybody's not doing this, and it's less consistent. It brings out the individual differences. You can't do the same. You have to go, okay, how far did you get yesterday? We're going to start with that. And so it's been a challenge to do this in a way that I'm used to doing research, but we tried, and I think I've learned a lot, and we're continuing to learn a lot about cognitive complexity and neuroplasticity and problem-solving ability in these animals. So it's been fun.
Are you going to try to continue layering skills on top of, so far they're steering and moving. Are you going to try to look for how complex their behavior can get, or are you going to take this further?
Right now we're interested in, we want to collaborate with a colleague who's looking at what aspect of neuroplasticity is important for this. So there are ways with some of our genetically engineered animals of suppressing neuroplasticity to see if they can learn it at the same rate. So learning more about the limitations of neuroplasticity and the important role it plays in this type of task could be very informative. So, my colleagues, and their defense, they've been quite supportive of this innovative little bit of research that we've done. So I think that we can try to pick apart what is important for learning these complex tasks, and maybe not unlike all the technology I've tried to learn during, blended teaching and all this. Our environments, the context of our brains are important in learning new tasks. Now, you could be very critical of me after saying, oh, we need to look at all these natural behaviors, and then I introduce Fruit Loops and driving cars. But we drive cars and we learn behaviors that our ancestors didn't do. So as long as I put it in that box, in that category, that these are acquired skills, then I think it makes it relevant. But I'm still very interested in looking at authentic behavior, being respectful of the behaviors of the species that we're looking at. As I mentioned, we're looking at wild rats now. We're looking at raccoons and trying to track them in the field and see what they can do, and learning more about their nervous systems and the design to learn more about ourselves. So we just need to branch out in a lot of different ways to learn some of these critical themes about optimal functioning, healthy brains.
So it takes you about a month to train up a lab rat, as opposed, well, I guess the question is, how long do they maintain that forever, or do they forget after some months, or is this a skill they have for life?
That's a good question, and no one else is doing this, and we haven't done it. And then I will say that we have a group now, and because it's kind of a popular area, we try to keep them trained up. But like during the holidays, during a hiatus of the holidays, where we're not training, we're just taking care of them. It seems to be like riding a bike. They, even with 3 weeks behind them, and maybe they haven't done it, they seem to jump right back in the car and drive. So it seems to be somewhat long term. I don't know if we stopped and then tried 3 months later, and 3 months is like a long time for human years if you start converting that, if they could remember. We did try, as part of that published work, a lot of people have asked, Kelly, do they like it? Do they like driving the car? Okay, well, that's a head scratcher. How do I test that? I can't ask them if they like it.
Stop giving them Fruit Loops.
Right. So because we've made the reward, associated with the reward, it's hard to disentangle it. They like Fruit Loops. I can tell you that. They'll work to get those. So we started what we call extinction trials between the enriched and the two groups we had. Mm-hmm. And so they would drive, but we didn't give them any Fruit Loops. And the ones that were the better drivers and the enriched animals, they kept driving longer. I don't know if they're more optimistic. That gets us into a different kind of category. But people have looked at optimism in rats, or they had a greater sense of control or something, but they stuck to it a little bit longer. And the idea that they stuck to it without the reward maybe suggests that at least it wasn't negative, that there was something positive about it. But so you get this cutesy little protocol, and then as you're suggesting, there's a lot of very complex philosophical, kind of deep questions that you can use to kind of answer these that we haven't been able to with some of the other types of tasks.
A question that's perhaps not so deep and philosophical is, well, I imagine you're doing these tests with both male and female rats. Is that the case? Do you find the stress response or cognitive abilities, or do boy rats prefer driving cars, or are they socialized?
And the girl rats like the dolls.
Yeah.
Well, what you bring up, looking at sex differences and anything related to neuroscience or biomedical sciences, is incredibly important. And our National Institutes of Health now, in my research that's funded, I don't have funding for the driving rats. We're just doing that on our own. But everything you propose, it should be that you look at males and females. For so long, we only looked at males because...
Why is that? Are they easier to deal with?
It's a pain to look at females. I'm a female. I'm looked at mostly males. Now we're looking at males and females. But the female rats have an estrous cycle, like a menstrual cycle, like human females do. Mm-hmm. It's about every 4 or 5 days. And so their hormones, the reproductive hormones, and even it has an impact on the brain, the receptors of the brain that receive those hormones, that's in flux, and it's changing depending on what stage of this estrous cycle the rat is in. And so for us to be able to do research with the females, if we're doing a cognitive task or an emotional challenge or something, we have to be like a little rat OBGYN and do vaginal smears. Take a sample of the cells and then stain them, look at them to see what stage of estrus she's in. Okay, you've just introduced a lot of time to pick the animal up, take the sample, take it back to the lab, stain it, look under the microscope. And you've also introduced a potential confound. You can't do it exactly that way with a male. We can pick up a male and kind of poke around the anogenital area. So it was easier and more control, we would have said, to just look at the males. But that's unacceptable because the, neurochemical milieu.
Because at the end of the day, you're going to be giving pharmaceuticals to females.
Exactly. And even with humans, it's still more of a challenge to deal with all the reproductive messiness of females. And then there's the weight differences and such. So we absolutely have to look at males and females. And so What
about pregnant females? Sorry for interrupting you. What about pregnant females? Are they also different when you're?
We've done a good bit of research about how the maternal rat brain changes when she's pregnant, when she gives offspring, and when she gives birth. And those offspring, in an environment where there are the required necessary resources, food and not predators and such, those offspring are kind of like an enriched environment for the mom. It's new sounds and smells and tactile stimulation. And the brain changes and her behavior changes in very fast, very different ways. So yes, not only estrous cycle, but if an animal's pregnant, then that's a whole different category. The hormones are very different. And that's come up with, I keep mentioning COVID because it's on our brain. It's tested in males and females, but a lot of people are asking, what about if I'm pregnant or lactating? And those weren't part of the original, sampling, or even kids. So we're at different neurobiological stages, and there are differences. So for our best predictions and specificity, we have to look at control as many variables as we can, but absolutely males and females. What we have done in looking at all of our cognitive and emotional, we certainly haven't seen that, males are smarter than females. It's just little subtle things that in this task, maybe the females are a little bit more bold. In this task, maybe the males are a little bit more active. Stress responsivity, maybe the females are, more affected by the stress, and they're, if we look at depression,
is that because
the rates
in nature they have to look after is that in nature because they have to look after not only themselves but also
it certainly could be. There could be some evolutionary determinants of that. We still, with females being twice as likely to suffer from depression than males, that's one thing that seeing, with the animals that's conserved, that the sex differences. But you're right.
Is that true?
Yeah.
I didn't know that. Okay.
And looking at sex differences with different psychiatric illnesses, that's another piece of the puzzle, a clue. But I think you're right. There may be something about females and childcare and vigilance and being responsive and empathy and sympathy and being more in tune with offspring and their well-being that may contribute to this difference in the stress response. That's very adaptive in the short term to taking care of offspring, but maybe not so much in the long term. We know it's not in the long term. So females are more than just that, but they could have its evolutionary roots related to that.
Did you find, I don't know if you tested this, but were female mice who had given birth better in the car driving tasks?
So we haven't done the effects of pregnancy in the car driving task, but we have done other like cognitive mazes. And being a mom makes them more efficient learners. We've spent years looking at that. Their stress response is a little bit more efficient as well. Now, the female rat is lactating for up to 12 pups in the lab. So they need to be very efficient with their energy expenditure. So being stressed for long periods of time goes against, that's counter. They need to be a little bit more relaxed for the lactation and such. They're also more efficient foragers, and that's why they probably are doing better in these learning tasks. The pups are very vulnerable. They're like little pink, hairless, helpless things. So if she's spending hours off the nest finding food to feed this, this heightened energy expenditure that she has, those pups are going to be a snack for a frog or a snake or a cat or something. Mm-hmm. So she needs to be very efficient and fast, get off the nest and get back to protect the offspring. And rats are single mother family models. The dads are just really sperm donors. So you have different family models with different species. So there's a lot on that female rat. So it's just really amazing some of the transformations and adaptations she makes quickly to be able to, yeah, to just put so much energy into taking care of these other little smelly animals. And that's huge that nature has devised a way for other animals to care for them. And that is probably really important. And our, when we look at I think parenting was a huge evolutionary step for nurturing and empathy and sympathy and some of the things that we think are the best about humans and living, caring.
It makes you wonder why female or even male mice aren't always, it sounds like being efficient when it comes to obtaining resources would always be a good thing in terms of energy expenditure. So is the reason why mice aren't always that efficient because they would exhaust the resources in their immediate surroundings, or what's the reason
every
why?
situation, and you're going to have different cost benefit kind of ratios. So a female rat, a lactating rat, high up on the priority list, she's got this huge genetic investment. She needs to keep those pups living so that they can, not that she's consciously aware of this, but they're going to pass on their genes. But if you just had some animals are more reflexive. They're just going to go this shortest distance to get the food and then come back. So that is very efficient. But what happens if that food's no longer there and they haven't explored and they don't have a sense of what's out there in the environment? There's a fun book written, kind of a little parable book 20, 30 years ago, entitled Who Moved My Cheese, for business kind of execs, about if you just keep going to the same place to get the same rewards, that's great as long as the rewards are there. But you also need to explore. So we have this kind of notion of explore versus exploit. And in different times, you're going to need to do both of them. But if you only exploit, we know humans, you're going to exploit all those resources, and then you have death. So it's a balance. And depending on the type of brain you have, an animal and situation, and who you're trying to defend, that balance will probably change. And you're more flexible, adaptive, opportunistic animals. And I put a rat, I would say that we're opportunistic. Rats are opportunistic too. They're willing to go wherever we are, eat whatever we eat. Raccoons fall in that category. So the animals that can engage in more flexible behavior and are adaptive, you're going to see these change depending on what the needs are at that moment. So a sick animal is not going to do as much exploring. They're going to exploit to get better. And a lactating animal will too. But when they're healthy, they may look around more.
So I should think of this as a different mode rather than, this mouse is a better ability than that one. It's just they're acting to different stimuli. But going back to neuroplasticity, trying to read from, if I want to learn something in my own life, like what should I be doing in my own life? What can I learn from your research? Are there some activities I should be doing to improve my own?
Yeah, I need to remind myself of this as well. So in the animal research, if we can generalize, which to some degree we can, one aspect of neuroplasticity is this kind of neurogenesis, producing new cells, and running, putting rats in a running wheel is the number one way to increase those cells. So we may not be producing as many as we're in adult and later adult, but it also increases this substance called brain-derived neurotrophic factor, BDNF, and it's kind of like a brain fertilizer. And so we see higher levels of this in some of our enriched, engaging models with rats as well. So exercise is important. I don't know if you need to run or you can walk quickly. I don't think that just watching other people travel and exercise on the screen is probably going to do it. But with so much of our brain devoted to movement, moving our bodies is incredibly important. We know that stress reduces, chronic stress, as I've been talking about, reduces neuroplasticity. So anything you can do to reduce stress is probably good as well. I've liked to maybe something that's just relaxing, but also you can reduce stress by gaining a sense of control. And that's where my research with effort-based rewards. So during the pandemic, people have been talking about baking bread and cooking and doing other hobby. I think that's perfect. I think that we could write prescriptions to hobby stores for there's research showing that knitting, it releases, that is related to reduced depression, and repeated movement can increase serotonin, and knitting, if you're counting the stitches, it's predictable, you're not thinking about your problems, may reduce stress. If you're doing something with a group of individuals, there's another neurochemical called oxytocin that seems to suppress stress hormones when you're with people that you identify with and have positive experiences with them. So that's the examples of how we can keep stress low and engagement higher, changing the neurochemistry and these factors to keep our brains ready to change, and so that we're not in some long-term sickness, learned helplessness kind of mode, that we can do what we need to do there. And I think it's okay to be sad, especially when we lose a loved one. I have issues with, do we need to take an antidepressant when we're grieving a loved one? Is that depression? Does that protect us against depression? Or do we need to know that this is normal and we're going to work through it? I'm not making any claims about that, but it makes you think. And we've redefined when you can prescribe due to grieving. But I think that it's healthy to have a full array of emotions, but when they become chronic and dysfunctional, we need to look at that. So exercise, sense of control, hobbies, being around other people, challenging, maybe learning new things, because we learn from the enriched environments is putting new things in there so that we can be,
So an instrument
or a language. You're getting outside your comfort zone.
Beyond 30, you're not too late?
I said neuroplasticity from the womb to the tomb. So if you're alive, if you're breathing, you can still learn things, and we should be. We need to get out of our comfort zone and not exploit the neural circuits that are engaged, and explore there as well. It's just like the animals foraging, this explore versus exploit. And we can, the plasticity of the brain and all those connections that are hopping and forming connections we call synapses, trillions, numbers I can't even quite, count, these connections that can change daily. I mean, just with what we were seeing with when an animal, the female rat goes into estrus, her connections change, maybe because she's looking for love or out there looking for a mate or something. And then that changes with lactation. So yes, learning new things is going to force us to use new brain territory. So that's going to give us more options, so that when we're confronted with a new challenge, we have more, we call it a cognitive reserve or a brain reserve. We have more to buffer what may be in our, neurological destinies, like Alzheimer's disease or something like that. It's not a destiny.
So what I wanted to do is I wanted to wrap up by asking you three questions that are they're not directly related to your research, but they're sort of in the ballpark. Okay. So I was hoping, while I have you here, to take advantage of that. So these are a little bit left field. I apologize, but can you die of a broken heart?
Can I do what?
Can someone die of a broken heart?
Die of a broken heart.
I mean,
okay. Emotions and stress can be quite debilitating. And people who, there's evidence, I'm going to go off base a little bit, but it's called voodoo death. So people who have thought that a curse had been placed on them, it's been recorded that even though it wasn't, and it was a trick or something like that, that the emotion, thinking that they were going to die, kind of shut down some of their autonomic functioning. And so, emotion, so being very, very sad or depressed, one, if you say broken heart, that I assume you mean brain. Isn't that funny? I mean, the ancient Greeks used to talk about the heart being, well, the ancient Greeks went from the heart, Hippocrates, to the brain, but we still send we celebrate Valentine's Day and say, I love you with all my heart. Well, that's not very impressive, because the heart is not experiencing these emotions. It's connected to the brain, but there are emotional circuits and areas in the brain that connect to areas that can activate pain, like dental pain or stepping on having some injury. And so our psychological perception of pain can be very similar with tissue damage and something being very, very sad. So we experience pain in that way, but also chronic stress related to loss can disrupt immune functions and cardiovascular. So the stress could disrupt the heart and lead to sickness, illness, and such.
Do we have any idea, imagine you had a magic lever that you could pull and remove all depression and anxiety from the population. Do you have any sort of idea what sort of impact that would have on other disease? So the instance of heart failure, or do we understand what the links are?
Yeah, for a long time in medicine, the immune system was thought to be completely separate from the nervous system. And if you were in medical school, it'd be two different classes and books. But we've known for decades now, there's so much crosstalk between the immune system. So you have immune receptors in the brain and neurotransmitter receptors on immune organs and receptors in the body. So there's so much crosstalk. So if we could dismiss or get a handle on chronic stress, then that would probably lead to more healthy outcomes for immunological threats or diseases, cardiovascular disease, and such. But I want to make it clear that I'm not saying that we should dismiss stress altogether. Mm-hmm. That is like our immune system saying something's, it's like I was saying before, something's wrong. And, I don't know what it would do to us as a species if we didn't have stress and emotional pain. Would we care anything about anyone else? Would we have any charitable kind of notions?
Or would we act to make any changes in our life at all?
So stress, acute stress, is very important for our functioning and to tell us when something is not right. Chronic stress can lead to dysfunction and chronic disease. And that's what we need to be careful about. And we're vulnerable to that because of this complex brain that can make so many associations and generate new things to worry about that maybe aren't even reality based.
So my second question is, and again, this is, in some sense, I apologize for this question because, but I imagine when you're dealing with the various rodents that you're working with, they have different personalities. But at the same time, you want to see, if I give them this environment, how they're going to act on a general level. So has your research had any impact with regards to your view of free will?
That is a deep one. Yeah. Well,
you can answer it at surface level if you want.
I think that our brains are sculpted by our experiences. And so to the extent that our experiences bring us to a point, where we can make an informed decision, however you're going to define free will, that through our experiences and the neural networks that we've built and emotional regulation and such, that we can make a decision and then act that way. But I don't think we can do that outside of any influence of, our neural being as well. We can't, you can't just take that out of the brain. So some neuroscientists would say, well, no, that means that we don't have free will, but we have an ability to regulate and to direct. And I think with the knowledge that we can do that, coupled with the knowledge that we are the product of these experiences and that we can still change them, that there's some hint of individualism and sense of control and efficacy that dances around free will. Is that
Yeah, I suppose you could describe free will as being sort of an effective construction, even if you don't believe in it even if you are completely mechanistic about something, you can mechanistic. Sorry.
Well, I guess
So
if you look at it as kind of the individual kind of regulation as an emergent property of all of these 86 billion cells or such, that has properties that you can't really identify specifically in any individual cell. I kind of think that's what a mind is. It's dependent on these cells, but it emerges and takes on some qualitative differences just in the sheer numbers and processing ability of that. So that's a hard one for a neuroscientist to, I think about it though.
Well, it goes back to the start of our conversation when we were talking about the number of neurons that different species have and the qualitative versus quantitative abilities of different animals. So it's not so out of left field.
No, it's a good question.
The other thing I wanted to ask you about, are you I'm guessing you're familiar with John Calhoun and the Universe 25 experiments.
Yeah, the crowded.
So in light of your research, first of all, we should maybe I could ask you to give a brief overview of what those experiments were, because it's sort of out of my field. But in light of your research, I was wondering what your interpretation of those experiments is.
Right. So I'm not a strict student and read closely all John Calhoun's work, but he was working interested in ecological questions related to behavior, working with National Institutes of Health. And I do applaud him. He brought, we're talking about animals in limited cages, so he brought them out to what he called a utopia and built these pens in his yard, I think, and then maybe around NIH that he called Universe this and that. And again, he did say that it was a utopia, and he just let them keep breeding. And so they became very crowded, these pens. And he noticed he talked about the behavioral sink, I believe, of just kind of the collapse of rat society. If we even knew what rat society really was, where they were.
They ended up with all of them dying, right? In these experiments, they had 100% mortality.
Well, I guess I'm more familiar with, I don't know that there was 100%. I get you've obviously read that. I was thinking more about there are more like attacks and then death. I mean, there was more death. My guess is that most of the time, there's going to be even some higher ranking animals that are going to live, but it almost looked like pathological behavior. And then he translated and speculated about humans living in cities and could see rapes and attacks and not taking care of offspring and such. So my interpretation, without knowing the fine details of Calhoun's work, and I know it got a lot of attention, is that I applaud him looking at behavior outside of a laboratory cage. I think that's important. But I don't see that environment as a rat utopia, because there was one important resource that they didn't have, and it was space. Once they were so crowded, they didn't have space. And we were just talking about how travel is important. And so it wasn't even really natural. In a more natural habitat, they have tunnels and they bury under the ground. And they do travel. They some of the research says 20 meters a day. They don't go miles. They could, but they don't. But they're going different places. So it's not like hundreds live in one big pot or something like that. So my interpretation would be that that's interesting what he did, but I think it was more stress related, what he was seeing. It was just they didn't have the resources, the nesting, the space, and it became very, very stressful. I would imagine that would have been a very stressful environment. Now, are cities stressful environment? I mean, if we're going to talk about cities, and we've talked about the city and the natural and the country rats, it's different. You have more resources in cities, but there can be, if there's more stress, then that's not going to be good. And going back to where we started with psychiatric illness, there is some research, such as with schizophrenia, that shows higher rates with cases of schizophrenia in more densely populated urban populations. But that's a very complex question or problem from an epidemiological point of view, looking at healthcare and social connections and resources and safety. So I don't think we could just say, well, living in cities is going to make us pathological, or being around people to a certain degree could. But stripping our resources and being stressed in the midst of the density of population, that's probably going to have a lot of negative outcomes. So I interpret it more as it being a very stressful environment, what he created at the end of the day.
The reason why I was curious about asking you was because in your research you deal with these trust fund and then worker rats. And then, in his experiment, the rats had everything in terms of food and water. They had that in buckets. So I was wondering if these rats may have been less resilient to the overcrowding, for example.
I think so.
Was the utopia? Was the utopia?
That's a very good point. I don't think it would be different than the laboratory cages where they're given their food and water. But you're right. The way we think about prosperity as humans today, I think it's very different than the way our brains think about prosperity. So he gave them all the food and water they wanted. They didn't have the space. They had each other. So they didn't have to have a sense of control about finding things, and if there's something to the effort-based reward and self-efficacy, which is hard to say for a rat. But when we think about, it makes us think about prosperity in our own lives, where we think, the more accomplished we are, the more, we can pay somebody to cook our food and to, these days, shop for us and to build our houses and to make our clothes. And you go just back, 100 years in this country, the pioneers, they were doing all this stuff. They were planting and canning, and they were controlling so much of a part of their lives and their resources that had to enhance their sense of control over the world. And they didn't know everything that was going on everywhere to worry about it. So stress was going to be lower. So prosperity today, where we want to sit in our fine houses and have everything done for us, that's not probably very good for our brains. And so in that way that he gave them all that they needed in their high-rise apartments or whatever, that's different. They were walking all over each other. So that's, and the smells and such. But I wish we could redefine and rethink and recalculate prosperity in the sense that we never need to give up the sense of control, even though I know we're living in different times and we have to, buy in and use like we're doing now, but not forget that there's value in making that bread or whatever hobbies or sense of control, learning new things, exploring new things. That's what brains evolved to do. So if we put it in a nice little package and have everything done for it, is that why we have depression? Is that why depression rates are higher? I would argue it's a factor. Could be other things as well.
So if you had your dream scenario unfolding in the coming decades with regards to mental health and depression, what would that look like in terms of treatment from what you've learned in your research?
Yeah, I know it's messy to talk about lifestyle, but I think we need to really explore the neurobiological factors in the context of lifestyle. So as we learn about how hormones change when we're exercising and engaging in this hobby, and when we're to have positive social interactions and learning new things, that can be part of a preventative type of or healthy lifestyle that we're more aware of and sensitive to. So like play, we think that maybe we're doing our kids a great service by taking all that rough and tumble playground stuff out of the classroom, because we want to focus on attention and cognitive and all of this. But play is incredibly important for developing brains, with mammals at least. And so there are models in other countries where every 90 minutes kids go out and do this rough and tumble play. And rats engage in this. It looks like two little kids pinning each other down and pulling their hair, or its tail in their case. So if you restrict play from rats, there are negative consequences. We haven't even done those studies. We know that children raised in institutions and impoverished conditions, they're not doing well. But we need to look at things like that, play. And I wasn't kind of kidding that maybe we could expand treatments into more behavioral treatments, such as hobbies and classes. And some of the early psychiatric treatments in this country were initiated by the Quakers, actually, but it would be kind of a retreat that you would go to, and there were classes, and you had to get up and get dressed, and grooming and learning about health, and very respectable. And the records and data were collected in a different way then, but it was written that more people were able to leave and live an independent life after that. It's less convincing, well, that in contrast to institutions decades ago was up to the 1950s is very scary, which was impoverished. So we don't want that. Mm-hmm. But we don't have evidence of our treatments now, such as pharmaceutical. Maybe a drug works and you don't have, you're not depressed, but you have to keep taking that drug. Or you're not able to do the things, you're not depressed, but you're not able to work in the same way. I want to raise the bar to get people back to productive, meaningful lives. Not just I'm not having suicidal thoughts anymore, or I'm not clinically depressed, but I'm back where I need to be. So I think we've just got, I think the lifestyle and looking at raising the respectable, when I say knitting, people go, well, that's just soft science to say that knitting or woodworking, that has any kind of psychiatric value. But let's think of all those connections, all the activation of the brain as you're planning a task, you're interacting with others, you're engaging in this task. That's incredibly activating and engaging for a brain, more so than putting a pill out of the context of real life. Now, again, I'm not saying that pill isn't necessary, and certainly don't stop taking them if you are. But we need to expand the way we think and what we can learn about neurochemistry and neuroactivation, so that we can do things earlier and that we can respect things like play and training and learning and being able to fail. We learn so much from that. So embracing some of these things and working, cooking, some of the things that maybe we've kind of written off as being, we don't need to do that anymore.
Or below us or something.
I would talk about the contingency conundrum, in the sense that our brains, these human brains, they're so innovative, and they got us in a situation where we can communicate in different countries via Zoom and do all these, digital and all this higher level scale computations. But that same brain has created that, we've created the technology to keep us from doing the very things that made our brain so good at doing that. So we're kind of in
our brains are helping us be as lazy as possible.
Learned helplessness or something. So we don't want to be in this downward tailspin where we're kind of too smart for our own selves. We need to still be in these uncomfortable problem solving, types of situations for our brains to be healthy. Maybe it's more fun to just sit and binge on Netflix. We're not stressed, but we're not doing too much for our neural networks, I don't think, doing that. So we need to respect that just as eating right is important for our health, engaging in a certain kind of balance of activities, and that's cognitive and behavioral, is going to be important if we want these brains to last as long as our medical information is allowing our bodies to last. We've got to catch up in that way. So it's probably not going to be in a pill, and it's not going to be easy. So we have to work at it. But if you pull a muscle, you can't just take painkillers. You got to start working that muscle again, right? It's not fun. So we've kind of created a mess, a conundrum that we need to be aware of. But short answer is to embrace and respect the healing power and the health related aspects of many different types of behaviors and endeavors, the way we do other medicinal therapeutic treatments like drugs and surgery and such.
So get out and use my hands.
Use it or lose it. Use
Play
it or
chess.
lose it. Yeah. And the hands, so much of their brain is connected to our brain, allows our hands to move in very sophisticated ways. In a similar way that the raccoon does is another reason I'm very interested in the raccoon. They've got those forepaws going. So, right. Engaging with our hands so that we feel it, that's different than just, typing is one thing, but it's just not the same as some of the other, the way our ancestors with planting and weaving and hobbies. And so we need to kind of get back to some of those maybe basics of our ancestors as we're doing all this, embracing and benefiting from technology, to not forget, just like we don't forget that we need to eat certain nutrients.
Escaped Sapiens.